Hi there, and welcome to this tutorial on GCSE Biology for the AQA Specification, focusing on reproduction and in particular on DNA structure. I'm Shumina from StudyMind, where we help you revise GCSE Biology with our helpful video tutorials, tailored to your subject, your specification, and to you. If you're new here, make sure you click the subscribe button. Whilst you are watching, please leave any comments below if you're unsure about anything, and let us know if it's your first time watching our videos so we can send you our free revision materials. We also have helpful timestamps below for each part of the video to help guide you through the specification. So let's get started. Hello there, and welcome to this tutorial on DNA structure. So in previous tutorials we've had a look at sexual and asexual reproduction, meiosis, and then discussed the advantages and disadvantages of these. We then moved on to have a look at a very introductory tutorial on DNA and the genome. And now we're going to have a look at DNA in much more detail. So this is quite a chunky tutorial, as you can see by the very long list of learning objectives and also specification points. So I would really suggest you do go through these specification points at the end of the tutorial and just tick them off if you do understand them. And if you don't, then maybe rewind to that part of the tutorial that will then explain it for you. Right, okay, so let's go. So this is DNA. Okay, so we have our phosphate backbone on the outside, and on the inside, shielded from the external environment, are our bases. Now, the structure of DNA is really paramount to its function, and this diagram is going to make a lot more sense to you by the end of this tutorial, so just have a look at it for now. You'll understand a lot more at the end. So, as we know, DNA is a very important molecule in the body, and it's also very complex. So you only need to have a simplified understanding of DNA for the GCSE syllabus, but it is still pretty complicated, so do stay tuned. I think this video will really help. So DNA is made from 4 different nucleotides. Nucleotides themselves are made of different structures. So we have our DNA, which is made from nucleotides, which themselves are made from different structures. And one of the structures that they're made of is our 4 nitrogenous bases. So it will contain each nucleotide will contain one of these bases. So this is just an outline. Now let's have a look in a little bit more detail. Also, purines must bind with pyrimidines. But that probably makes absolutely no sense to you at this point, and that's completely right. Let's just go on and have a look at this, and it will make much more sense once we visualize it. So this is our nucleotide. So we call nucleotides the monomer of DNA. Okay, if I'm just to write that word for you here. Monomer. Mono means one. It's your basic subunit of DNA. So your single unit of DNA is your nucleotide. Your monomer of DNA is your nucleotide. These monomers are put together to form a polymer. So many monomers make a polymer. Poly means many. Okay? Really simple. So our nucleotide is our DNA monomer. The nucleotide itself is made of 3 different structures that you need to know about. So it's made up of a sugar, a phosphate group, and a nitrogenous base. So the sugar in DNA is deoxyribose. Oopsie, did not mean to draw on that. So you probably know many different types of sugars, like glucose and fructose. Well, here we're using deoxyribose in DNA, okay? Now, the deoxyribose sugar binds to a phosphate group and to a nitrogenous base, as you can see. So it's bound to its phosphate group and its nitrogenous base. Now, it actually binds to another phosphate group on this side, and that phosphate group belongs to another nucleotide. So imagine this, or you don't actually have to imagine it because I believe it's on the next slide. There we are. So we have our 1 nucleotide here, a second nucleotide here, and can you see, if I were to divide it up around here where that phosphate linkage is, this is nucleotide number 1, this is nucleotide number 2. Now, this deoxyribose sugar binds two phosphates. One is its own phosphate, one is the phosphate of the neighbouring deoxyribose sugar, and this is what allows our two monomers to form a polymer. So imagine this whole structure repeating itself again and again and again. These are each going to link up to identical subunits, and that's going to form our polymer of DNA. So do you see how we have this little monomer here that's able to then join up to the phosphate of another identical monomer to form eventually a polymer as it joins to more sugars, or more nucleotides I should say. So this is how the DNA forms a chain. Now there are 4 nitrogenous bases, so if I just flip back to the other side, this is what I mean when I talk about our base. And this is really important in DNA, okay? Because do you remember on that first slide when I showed you the overall zoomed out structure of DNA, you could see the helix shape of it, so it kind of zoomed around like this, didn't it? That's really not a good diagram, but you get the idea. And what I'm trying to draw is, we saw that there was a phosphate backbone on the outside. Perhaps if you flip back to that slide at the end and have a look, we saw there was a phosphate backbone to the spiral, and on the inside were the bases. Okay, and the bases are the really important parts here. We have our 4 nitrogenous bases, which are adenine, or A, thymine, T, cytosine, C, and guanine, G. And we just write them as ATCG because who has time to write out their full names and remember the correct spellings? So you're allowed to refer to them as ATCG, but make sure you also know their full names and how to spell them. So the nitrogenous bases bind with one another to join nucleotides together. So we saw previously that there is a bond with the phosphate groups and the nucleotides, but it's the bases that bind to each other that join the nucleotides together a lot stronger. Okay, so if I just try and draw that out for you here. So this is our deoxyribose sugar that I'm just drawing. It's attached to a phosphate. It's also attached to a phosphate of another deoxyribose sugar. Okay, so that's our other deoxyribose sugar going down here. And finally, it's also joined to our base. Now, what I'm saying is it's not the phosphate bond that is the stronger one here. It's the bonding of our bases. So it's this bond that is the one it's all disappeared, but it's the bond between the two bases that is what we need to worry about, okay? It's the bond between these two, and that's the stronger bond, and that's what binds DNA together really strongly. So adenine and guanine are purines, whereas thymine and cytosine are pyrimidines. So that's just a method of classification of the bases, okay? But just commit that to memory. The reason why that's important is that purines bind with pyrimidines. Therefore, adenine must bind with thymine and cytosine must bind with guanine. And so it's always A and T binding and always C and G binding. And you need to commit that to memory because that will be tested really easily in the exam. So any other types of binding that do occur by an off chance have awful consequences to the body. So people with any other sort of binding apart from AT and CG are not doing so good. Like that's a very, very severe physiological abnormality there. Now, each amino acid is coded for by a sequence of 3 bases. So there are 20 amino acids, and each one has a few possibilities of sequences of bases that build it. Now, the code that defines which sequences of bases for which amino acids is known as the triplet code, because I've said already that it's 3 bases that code for a single amino acid, and we call this the triplet code. So you can see here we have our bases, and we group them in groups of 3, and that's how it's read. So we call a group of 3 bases a triplet code or a codon, and that is going to code for the amino acid, the relevant amino acid. So this triplet code has 3 important features. So number 1, it's universal. So all organisms use the same codons, so the same sequence of 3 bases to form proteins. Number 2, it is degenerate, and this means that several codons can code for the same protein, and we'll see that on some slides to come. So this shields organisms from mutation to a certain extent because it means that if there is a mutation in a codon, there's a chance that it could still code for the same protein, or the same amino acid, is what I mean to say. Also, the code is non-overlapping, so that means that it has to be read in its sequence of 3 bases before it moves on to the next 3 bases. So if anything is kind of removed from any of those bases, then that means the code is going to be read differently. Because if we have base number 1, base number 2, base number 3, base number 4, base number 5, base number 6, base number 7. So this here is part of triplet number 1, this here is part of triplet number 2, and base 7 is in with triplet number 3. Now let's say there is a problem here. Base number 1 gets deleted, which means that now I'm going to start reading from here. So the triplet's going to go like this, and it's going to produce a completely different protein at the end of it. Okay, so this is why I say the triplet code is non-overlapping. It has to be read in the sequence that it came in, 3 bases at a time. Now, you've got to learn these 3 features of the triplet code because they are often tested and they're really easy marks. So just learn those 3 buzzwords on this slide over here. So universal, degenerate, and non-overlapping, and make sure you understand what they mean too. Okie dokie. So now let's move on to the next specification point. So DNA's biggest process is protein synthesis, because this is how we allow for growth of our body and also repair. So DNA provides a template upon which proteins are coded for. So there's a bit of confusing terminology coming up, so don't be afraid to take it slow and I'll try and explain as much as possible. So transcription and translation. So protein synthesis is the process by which the base sequence found in genes on DNA is used to make polypeptides. Polypeptide is just another word for a protein. So it occurs in 2 major phases. We have transcription, in which a strand of mRNA is synthesized from a particular gene template, and we have translation, in which the mRNA attaches to the ribosome, which recruits tRNAs carrying amino acids in order to make a polypeptide. Now that probably sounded like a whole load of gobbledygook, but don't worry, we're going to go on to and explain this in a lot more detail. So first of all, RNA. So RNA is a ribonucleic acid, and it's an important part of protein synthesis. And it's really important because DNA always has to stay in the nucleus, but somehow we've got to end up with proteins in the cytoplasm. So RNA is almost like our medium. It's like our transitionary state between the DNA and the final protein that we get. And this is because RNA, in the form of mRNA, which stands for messenger RNA, is actually a copy of the DNA. So that's copied within the nucleus. It then leaves the nucleus and moves to the ribosome, which is found in the cytoplasm. And the ribosome is a cytoplasmic organelle that is responsible for protein synthesis. So this copying of the DNA from DNA into mRNA is known as transcription. So you can see here we have our DNA in the nucleus. This is the section that's being copied, and we can tell because it's slightly unravelled, it's DNA. And we have a copy of the DNA that's being made in the form of mRNA. Now notice here these bases are not an exact copy of the DNA. They're in fact the complementary base of the DNA's base. So remember we talked about how purines pair with pyrimidines, so it's always A and T and C and G. Well, in mRNA we have U instead of T, but don't worry too much about that right now. You don't need to learn these details. But my point is that mRNA is just a copy of the DNA, but it's in the form of its complementary base pairs. So these bases all pair up complementary to these bases. And this forms our mRNA. Our mRNA can then move out of the nucleus into the cytoplasm, leaving our DNA nice and safe in our nucleus. So it works perfectly, so our mRNA is trafficked off to our ribosomes. So the mRNA reaches the ribosome, where it then recruits tRNA, which actually means transfer RNA. And this tRNA, just like how the mRNA was complementary to the DNA, the tRNA is complementary to the mRNA. And so that means our A's and our T are binding to our T's and our C's are binding to our G's. And the tRNA molecule itself carries amino acids with it. So on one end it has the complementary bases, and on the other end it has our amino acids. The amino acids then bond with one another and polypeptides are formed. So this part of the process is known as translation, whereas transcription was the mRNA DNA part of the process. So you can see here we have our mRNA, this is our ribosome, the tRNAs come along. On one side it's binding in a complementary pattern to the mRNA, and on the other side it's recruiting amino acids which are going to join up to one another and form a polypeptide chain. Okay, so in conclusion we have our transcription going on here, where our mRNA is copied from DNA. And we have our translation that occurs in the cytoplasm. So your examiners might ask you to describe one of these stages, so make sure you understand what is going on. Just remember that we go from DNA to an exact copy, a complementary copy of that in the form of complementary base pairs on the mRNA. The mRNA migrates to the ribosomes in the cytoplasm, and tRNA then pairs up with the RNA also by complementary base pairing. The tRNA has amino acids that are associated with it, and therefore recruits these amino acids to the site of the ribosome, which join together and therefore form the specific sequence of amino acids required to form a specific protein. Okay? Now, this therefore means that the order of the bases in the DNA ultimately determines the order of the amino acids that are recruited with the tRNA. And remember, it's this order of amino acids that then determines the specific polypeptide made. Because remember, our primary sequence of our protein, which is covered in the protein tutorials, is what determines our ultimate structure of our polypeptide. So the structure of the DNA influences what protein is made. So the way in which the bases are paired here influences this amino acid sequence here. Now, once these protein chains are formed, they are folded in unique ways so that each protein has its own unique shape. And this is imperative for enzymes, for example, because, as we know, they have a unique individual active site. And these active sites have to be complementary to their substrates. So actually, the exact amino acid sequence in the protein determines this active site structure and therefore allows for binding to the substrate. So you can see why DNA is important. So the expression of proteins can be altered by switching genes on and off. And DNA itself does not all of it codes for proteins. So DNA is separated into coding regions and non-coding regions. So it's the coding DNA, that's what we looked at just now, and that's what directly determines the proteins formed. Non-coding DNA, on the other hand, has a different function, as it works to turn on and off genes. So therefore, I like to think of non-coding DNA as being the controlling regulatory sector, and if it decides to turn off a section of coding DNA, the protein produced would be different. And this is actually really important, because remember that every cell in your body contains an exact copy of each other's DNA. So every cell contains all of the DNA in the body. But, your liver cells produce insulin, for example, but your muscle cells don't need to produce insulin. Or, your cardiac cells, for example, they have an electrical pacemaker within them, but your other cells in your body don't all present with that either, do they? So each type of specialised cell in your body does these specialised functions, even though at the end of the day, every single one of these specialised cells contains all of the DNA. So therefore, this shows that genes are being switched on and off in order to regulate which cells are producing which proteins, which means that ultimately our liver cells can uniquely produce insulin and our muscle cells can uniquely produce ATP synthase. So therefore, different cells have different genes turned on and off, and that's controlled by these non-coding regions of DNA in our body. So next tutorial point, so mutations. Mutations are random changes in the DNA sequence. And we saw that the DNA sequence is paramount in coding for a particular polypeptide. So if we have a change in DNA sequence, we may have a change in polypeptide. And this can lead to non-functional proteins, as we saw with the enzymes. But actually, mutations occur all the time because our cells are replicating at such a crazy rate. And the majority of mutations don't cause profound changes in proteins. So this is for many reasons. So first of all, we saw that the triplet code is degenerate, which means that in many circumstances, one base may change, but that triplet will still code for the same amino acid. So this means that the right protein will still form. So this is an advantage of the triplet code being degenerate because the codon may actually end up coding for the same amino acid, because many amino acids are coded for by more than one codon or by more than one triplet code. So therefore the functionality of the protein may not be affected. So you can see here, for example, this codon here, or triplet of bases, is coding for arginine. I'll call this codon A. This is codon B. Codon A is GCA, codon B is CGT, but they're both coding for arginine. So this is why we say the code is degenerate. However, in cases such as enzymes, even small mutations can lead to changes in the active site, and this can make them non-functional. So this could be problematic in DNA that codes for structural proteins, because if a sequence for a key structural protein is altered, it can lead to big problems in the body. So you can see how changes in the specific sequence of DNA can cause big disadvantages. Now, this just gives you a further example. So if we have our normal amino acid sequence being produced, we have our enzyme with its specific active site. If it has a mutation where this leucine has mutated into an alanine, then we're going to have a different shaped active site. Now, mutations themselves can occur in three ways. So we can have deletion of a base, which just means losing a base. So you can see here, this base, G, guanine, has been lost, so it's been deleted. So this can often lead to a frame shift mutation, which can cause great issues. And by frame shift mutation, I mean that when this code is read, it's now going to be read like this, as opposed to like this. And therefore we're going to change the ultimate polypeptide that's coded for. Another type of mutation is an insertion. So this is just exactly as what you would expect with an insertion. An extra base is inserted into the sequence. So here we have a C put in front, I didn't need to draw on that, a C put in front of the guanine here, so a cytosine in front of the guanine. A substitution mutation is when a base is substituted into the sequence instead of another. So you can see here, alanine is in the place of guanine. And remember, this is affecting the ultimate protein that's formed because our DNA is read in our triplets. So here we're going to have a change in codon. Okay, so this codon is going to be different to this one, because this sequence of three bases is different to this sequence, and therefore it may possibly code for a different amino acid. But remember, there is a chance that they actually both end up coding for the same amino acid because the code is degenerate, as we saw on previous slides. So please make sure you learn these different types of mutation, these 3 different types: deletion, substitution, and insertion, because they can easily come up. So now let's have a look at a practice question. So below are 5 statements about DNA. So you need to select which statements are true. So A, DNA is described as degenerate because more than one triplet sequence codes for an amino acid. B, an insertion mutation can cause a frame shift mutation. C, multiplication is a form of mutation in which a base is multiplied to have 2. And D, the first stage is protein synthesis. The first stage of protein synthesis is protein translation. So pause the video at this point, scribble a true or false next to each of these, and we'll go through it. Okay, so first of all, this first statement is very true because we have already discussed that more than one triplet sequence of bases may code for the same amino acid, as we saw with the arginine on a previous slide. Statement B, an insertion mutation can cause a frame shift mutation. Yes, 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 because remember, our DNA sequence is read as triplets. Multiplication is a form of mutation in which a base is multiplied to have 2 of it in a row. Never heard of that. Sounds like a good idea, but unfortunately it doesn't happen in our body yet. And D, the first stage of protein synthesis is protein translation. No, that's the second stage, because remember, protein transcription, the stage in which DNA is translated into mRNA, occurs first, and then translation occurs when we have the mRNA on the ribosome recruiting the tRNA, which also has associated amino acids, and they then join together to form our polypeptide chain. So really simple, a long tutorial though, so do go back and rewatch, perhaps as you go to bed tonight, and maybe get these buzzwords into your head like translation and transcription and so on. But well done for today, and I'll see you for the next.
Thanks for watching this free video from StudyMind. If you liked this video, make sure to subscribe to catch our newest videos by clicking below, and leave a comment on a topic you'd like a video on. Click here to watch more videos in our series for GCSE Biology, or visit our website studymind.co.uk for free past paper compilations by topic and specification.